Hello and welcome to my channel. I am Dr. Sandeep Hatwar, pediatric and neonatal surgeon. In combination with Icon IVF and Fetal Medicine Center presents to you my series on human embryology made easy. This is my second video on gastrulation. In my first video we have seen the beginning of gastrulation with the formation of primitive streak. This primitive streak is formed on the 15th day of fertilization and it is very easy to remember because it contains 15 letters. If you have not watched my earlier videos, then please watch them. The link is given in the description box. And also please subscribe to my channel to get more updates on human embryology made easy. This is a recap of previous video. Here we are seeing a bilaminar disc with a primitive streak, which regresses at the end of third week of development and completely disappears by 26th day. Primitive streak is a primary organizer and it induces the formation of notochord and intraembryonic mesoderm. The prochordal plate, primitive streak and the notochord determines the cranio-caudal axis, the right and left side of the embryo, and also the dorsal and ventral aspect of the embryo. Gastrulation is the process of formation of trilaminar germ disc. Today we will discuss the formation of intraembryonic mesoderm, then the differentiation of intraembryonic mesoderm into three different types: the paraxial, intermediate and lateral plate mesoderm, and subsequently formation of intraembryonic xylem, then the formation of notochord, and finally the formation of neural tube. In earlier video we have already seen the formation of ectoderm and endoderm, and now we will see the formation of intraembryonic mesoderm. In earlier video we have already seen how the cells of the ectodermal layer they proliferate and migrate towards the primitive streak and fall like a waterfall through the primitive groove and replace the cells of the hypoblast. These cells they continue to divide even after replacing the complete layer of the hypoblastic cells into endoderm, and they continue to fall through the primitive streak. This is a three-dimensional view and you can see that these cells are now occupying a space between the ectoderm and the endoderm. Thus a new layer is formed between the blue color ectoderm and orange color endoderm on the right side like this, then on the left side like this, and also in the region of the connecting stalk. This is the connecting stalk, but this layer will not be formed in the region of cloacal plate and also in the region of notochord here and in the region of prochordal plate. This new layer is called as the intraembryonic mesoderm, and this is how it grows. This blue layer that is growing is the intraembryonic mesoderm, and now I have replaced the color with this color so as to avoid the confusion. Now we have a trilaminar disc. Now next is the formation of the notochord. To understand this, we will take a sagittal section of the developing embryonic plate. This is the sagittal section, and in this is the precaudal plate. This is the area of the primitive knot, and that is the area of the primitive streak. Now as you note that as the cells of the primitive knot, that is the Hassan's knot, they divide, multiply and fall through the blastopore, simultaneously there is a disappearance of primitive streak. Now watch here, as the notochord is developing, the primitive streak is disappearing. Initially a solid cord of cells is formed. Then the cavity of the blastopore extend into the notochordal process and convert it into a tube. The blue cells forming the floor of the canal intercalate with the orange cells, and then the floor breaks and a temporary connection is formed between the amniotic cavity and the yolk sac. This is the zoom out, and that's the amniotic cavity, and below is the yolk sac. This temporary connection allows the nutrient from the yolk sac to diffuse into the amniotic cavity so that it can provide nutrition to the developing notochord and the neural plate. Now watch the entire process again. The primitive streak disappears, a notochord solid cord is formed, then blastopore extend and convert it into a tube, then the floor cells intercalate with the orange cells to form a temporary communication, and then the floor cell again rise up, and then a solid cord is again formed, and this cord then subsequently becomes J-shaped, and that is called as a definitive notochord. Only a part of it persists in the humans as nucleus pulposus and apical ligament of dens of the axis vertebra. Now this is how the primitive streak closes from cranial to caudal end, and that is the primitive knot or the Hassan's node and a blastopore. and through the blastopore you can see a notochord like this, a solid cord of cells. This is the dorsal view of the embryonic disc. Now let us remove the upper ectodermal layer of the embryonic disc to see how the notochord looks like, where does it occupies a space. This notochord occupies exactly the center space, that is the axial space, and therefore it is also called as the axial mesoderm. Now watch carefully the region of the prochordal plate and the cloacal plate. This extra layer of the mesoderm, which is formed during gastrulation, does not occupy these two spaces and also the region of the notochord. That is the differentiation of the mesoderm subsequently to form paraxial mesoderm, intermediate mesoderm and a lateral plate mesoderm. The cavity of the intraembryonic coelom is subsequently developed in the lateral plate mesoderm. Now all these processes are happening simultaneously and therefore we are discussing them together with gastrulation. The region of the embryonic disc, which is cranial to the prochordal plate, there the two mesoderm from the right and left side, they fuse together to form the septum transversum, and this is the region of the connecting stalk where the mesoderm extends into the connecting stalk caudal to the cloacal membrane. Now let us again understand the development of notochord in transverse section in this region of the embryonic disc. Thus this is how first a primitive notochord and then finally a definitive notochord is formed. At the same time mesoderm is also differentiating. Let us watch all this process in slow motion. At first, the blue color cells of the primitive knot proliferate to occupy the space between ectodermal and endodermal layers to form a solid notochord. Then the cavity of blastopore extend and convert it into a tube. The blue cells of the notochord tube intercalate with orange cells of the endodermal layer and the floor breaks to establish a temporary communication between amniotic cavity and yolk sac. Later, the floor cell again rise to form a tube, which then converted into a solid rod of cells becomes J-shaped and called a definitive notochord. At the same time, we have seen that the mesoderm is appearing on either side of the notochord and getting differentiated into three different types: the paraxial, intermediate and lateral plate mesoderm. Also simultaneously a cavity was appearing in the lateral plate mesoderm, and that cavity is called as the intraembryonic coelom. So this is now the dorsal view, which shows a blue color ectodermal layer and the lowest orange color, that is the endodermal layer, and in between is the mesodermal layer. Now next is the formation of neural tube, and that is the process of neurulation. Now in this process, the cells which are overlying the notochord, that is the ectodermal cells between the prochordal plate and the Hassan's node, they proliferate and becomes differentiated into a different type of cells, which I have shown here as a yellow color, and that this forms a neural plate, and subsequently the neural plate folds and forms a neural tube and gets invaginated inside the two layers, ectodermal and the endodermal layer. At the same time, the embryo is also elongating and the body somites are also being formed at the lateral part neural plate, and then they fuse in the midline, and the neural pore, that is the anterior neural pore, and the posterior neural pore, that is the last part to fuse. At the same time, the embryo has folded, and that we will discuss in the next subsequent videos. Now we will again see the development of neural tube in transverse section at this level. Now this is how the yellow color cells, they fold inside and form a neural tube. Let us see in slow motion. At first, the ectodermal cells covering the notochord become specialized to form a neural plate. Then subsequently, as the plate is folding, at the edges the neural crest cells appear, and then once the folding is complete, the neural crest cells, they separate out and they get trapped between the ectodermal layer and neural tube. Also see the changes taking place in the mesodermal layer, especially in the paraxial mesoderm. There you see that the mesodermal cells which are floating in the stroma, they get themselves arranged at the periphery in a circular pattern. This circular arrangement of mesodermal cells will correlate to this phenomenon where the players, they arrange themselves before the match and then they decide the strategy, and then once it is decided, then they disperse and go back and take their own respective positions in the field. Similarly, these mesodermal cells, they decide what role they have to play, and then subsequently they form the dermatome, myotome and the sclerotome and differentiate into various structures. Now at this point of time, let us zoom out and see the structures in detail. Now this is what I am zooming out. That is the dorsal portion, and that is the amniotic cavity. This is the yolk sac. This planktopleuric layer of the extraembryonic and intraembryonic mesoderm, and the somatopleuric layer of the extraembryonic and intraembryonic mesoderm, the tropoblastic layer and the somatopleuric layer of the extraembryonic. The cavity which is highlighted black is the extraembryonic coelom, and this cavity which is highlighted black now is the intraembryonic coelom, and that is the point where the intraembryonic coelom extends cranially towards the precaudal plate. And this is how the extraembryonic coelom communicates with the intraembryonic coelom. Look at the direction of the arrow, how it is traveling. That is how the arrow going inside from the extraembryonic coelom into the intraembryonic coelom and then coming out from the other end. Now this is the top view or the dorsal view of the embryonic disc. The blue layer, the ectodermal layer, in between is mesoderm and lowermost is the endoderm, and that is where I am zooming in is the intraembryonic coelom. This is the intraembryonic coelom which is formed in the lateral plate mesoderm. Now zooming out again, showing all the details once again. The amniotic cavity, the yolk sac, the amniotic cavity covered and the yolk sac covered by the extraembryonic coelom. This is the tropoblastic layer, and this is how the intraembryonic and the extraembryonic coelom, they communicate with each other. And this is what we get finally. Now I am removing the ectodermal layer to show you what is there inside. This is how the layer is being removed, and this is the final appearance of the mesodermal layer. The various parts of the mesodermal layer, one by one, let us find out what are they. Now this is the region of septum transversum. These are the two dorsal aorta. That is the pericardial cavity, and these are the two pleuroperitoneal canals which connect the septum transversum with the future peritoneal cavity. This is the procaudal plate, that is the cloacal plate, and that is the neural crest cells on both sides, a layer besides the neural tube. The yellow color is a neural tube. So this was in a nutshell all about the process of gastrulation. See you in the next video, wherein we will discuss the folding of the embryos and formation of various body cavities and furthermore. If you haven't yet subscribed to my channel, please do subscribe it, and please do not forget to put your comment about this video. If you like it, please do share it. Thank you. See you in the next video.